Okay, so we are back here for this last little bit of the chemistry lecture. Again, I'll try to go through this as quickly as I can, but I know that some of you have not had this, for a while. So please keep in mind that I know that, for the chemistry section here, we have people that are at really different levels. You've got some people that, just had 181. This is all fresh in your minds. And then we have some people that haven't had this for, years. So again, the beauty of this, right, is that you have these lectures and you can kind of fast forward through the videos for, through parts that, you're really familiar with and you don't need the review. And then you can spend more time with the sections that you need more time. So that's kind of why I've spent a little bit more time on this chemistry videos than probably I normally would. And it's just to kind of cover all my bases. So like I said, if this is really fresh in your mind, feel free to kind of fast forward through some of the parts and move on. So really quickly, we left off with dehydration synthesis and hydrolysis. And these were kind of our two major reactions that we were talking about. Dehydration synthesis being our building synthesis reaction, and hydrolysis being our degradation reaction. So please keep in mind that these two reactions are going to be really important when we talk about these four major molecules that are important for the human body. And that is carbohydrates. We're going to talk about lipids, proteins, and nucleic acids. And to build your complex carbohydrates, your proteins, your lipids like your triglycerides, and your nucleic acids, we're going to use the reaction that occurs. I shouldn't say we, like we're not using that reaction, but the reaction that occurs for those molecules to build, those major molecules to be built, is dehydration synthesis. And then when we break down those molecules, hydrolysis is used. So that's why we talk about dehydration synthesis and hydrolysis. You're going to see these reactions. We gave you the example here where you're actually making a disaccharide. So you're going to see those reactions now hit you in each of these categories. So when we talk about carbohydrates, we talk about lipids and proteins and nucleic acids. You need to know that they are each going to be made up of these base monomers, and you're going to need to know what the monomers are. Remember, the monomers are the building blocks, right? So in this case, we'll talk about in just a minute, in carbohydrates, they're going to be our monosaccharides. And as we put those monomers together to build our polysaccharide, right, we're going to be using that reaction called dehydration synthesis, right? As we're putting together a monomer with another monomer, again, the dehydration synthesis is going to be the reaction that is going to occur. And that's going to happen again for carbohydrates, proteins, lipids, and nucleic acids. So for each of these categories, you're going to need to know what is the monomer, the building block, and then what are some of the, polymers. So again, for each of these categories, we'll kind of go over that. You need to know that dehydration synthesis was the reaction, the building reaction that put them together and made the polymer, and that hydrolysis is the reaction that's going to break that polymer up. Okay, so hopefully that kind of ties everything together here. Now, when we talk about carbohydrates, which is our first category, this is going to include our sugars and our starches. Everybody's familiar with carbohydrates, right? These are always the tasty things, right, that we think of. I love my starches and sugars, and of course, in moderation is the hard part, right? Because they tend to be the tasty things. So your carbohydrates are going to contain carbon, hydrogen, oxygen. Hydrogen, oxygen are in a 2-to-1 ratio, not super important for you to remember right now for purposes of our class. But you do need to know that the monomer for carbohydrates is called monosaccharide. Okay, so a monosaccharide, again, is just one single sugar. That's the basic smallest unit of a carbohydrate. And when we make something like a disaccharide, like we did here, right, we have to put two of the monosaccharides together. We put those two monosaccharides together via dehydration synthesis, pull out the water and build that nice covalent bond. between the two, then we form a disaccharide. Okay, a disaccharide, di meaning two, right, consists of two monosaccharides bonded together. Now, polysaccharide is even more complex, and that's so more than two sugars are put together. So many sugars are put together, and this is our polymer, right? So when we look at carbohydrates, a monomer is the monosaccharides, and we put those monosaccharides together to make our polymer, polysaccharides. So again, my funny cartoon with a potato, right, being starch, says you are very interesting, unique, and yes, complex carbohydrate. I can hear the laughter on this end right now. I know. I'm sorry, I can't help myself. I have to keep you going during these, these somewhat more dry portions of the lecture. So again, monosaccharides are these simple sugars. Of course, they're the ones that, are so nice and tasty to us. They're the ones that are really sweet. So again, what we love about cherries is that they have lots of fructose in them, okay? Or what you love about your, frozen yogurt is that it has lots of galactose in it. So these are the things, excuse me, as I'm getting over a cold here. So, but those are the things that you guys, like that give you that gives that food that sweet taste. So again, monomers of carbohydrates are monosaccharides. Oftentimes they can be pentose sugars or hexose sugars. Pentose meaning, again, it's going to form like a kind of like a pentagon type shape, and then hexose being more of like your glucose, going to form this hexagon shape. So some pentose sugars that we're going to talk about are going to be ribose and deoxyribose, especially in just a few minutes when we talk about nucleic acids. So nucleic acids include these very important monosaccharides. And then hexose sugars are going to be things like glucose. And so, of course, we talk a lot about glucose in the blood, right, in terms of your blood sugar. And so, again, these are just some examples of, some of these hexose sugars. Here is glucose and galactose are both hexose sugars. Fructose, deoxyribose, and ribose are these pentose sugars. And again, those are all monosaccharides. So when we put two monosaccharides together, okay, again, that dehydration synthesis of these two monosaccharides, then we can make a disaccharide. This is, again, a double sugar, and these disaccharides are typically going to be very bulky and large, too large to pass through cell membranes. Some of these important disaccharides are going to be things like sucrose, which is going to be your table sugar, right? Glucose and fructose bonded together make sucrose your table sugar. Maltose, lactose are all going to be examples of those disaccharides, okay? So again, and here's the example of this dehydration synthesis reaction that occurs, right, to make sucrose your table sugar, putting glucose and fructose together as your monosaccharides, again, and then dehydrating, removing a water to form that covalent bond between glucose and fructose, and you get sucrose and water. So this is just a couple of disaccharides. Here's sucrose is your table sugar. It consists of glucose and fructose. Maltose consists of two glucose molecules bonded together, again, via dehydration synthesis reaction. And then lactose actually equals galactose and glucose bonded together via dehydration synthesis reaction. And so some people are what's called lactose intolerant, and we'll talk more about that in terms of the digestive system. But what basically that means is that you're lacking an enzyme, which is going to be something we're going to talk about in a minute when we talk about proteins, but you're lacking an enzyme called lactase to break down this disaccharide. And so what ends up happening is if you can't break it down, that it can't be absorbed, right, through the membranes. And so it actually stays inside your digestive tract, and it can cause all kinds of problems. And, bacteria will end up, feeding on it and causing lots of gas and things like that. And later on in your digestive tract. So that becomes a problem for people that are lactose intolerant, that don't have the enzyme to break this bond, because if you don't have the enzyme to break that bond, then you can't absorb these individual monosaccharides. just to kind of help make it interesting for you in terms of the anatomy of it. Polysaccharides, again, are when you're going to put together a bunch of monosaccharides via dehydration synthesis reactions to form a chain, right, of or polymer of monosaccharides. We call that polysaccharide. Important polysaccharides are going to be things like starches. So, many of the foods that we like to eat that come from plants contain starches. And so we're going to have, these nice complex chains, right, of monosaccharides put together. And then we'll, of course, if we want to break down that starch, we have to have enzymes to be able to break down some of these bonds between the monosaccharides. And then there's glycogen, which is our storage form that's used by animals to store carbohydrates in a more complex form. So again, when we talk about glycogen, just like I was talking about earlier when I said a lot of times if you're training carbohydrate loading, you're actually trying to build up your glycogen stores. So that is our form. So here's glycogen again, polysaccharide formed from the dehydration synthesis of many of these monosaccharides put together. And you build these kind of chains of these monosaccharides built together, in this case, glucose. Molecules put together, and we store that glucose temporarily in the form of glycogen. Okay. Now these tend to be bulky molecules. They're not very soluble in water. And so, to be honest, they're quite heavy. And so we'll store our longer term energy temporarily as glycogen, but not for long purposes. And the reason being is that if we had to carry all of our energy storage in the form of carbohydrates like glycogen, we would just be massive. We would be so heavy because these molecules are heavy molecules. And so that's why we end up, when we talk about lipids in a minute, doing some of our more long-term storage of energy in the form of lipids. So again, glycogen is still, longer storage, obviously a longer term energy supply than glucose is. But again, it's not something that we want to store for really long term because it's a heavy molecule. So lipids. When we talk about lipids, these are going to be basically our fats and our oils, right? They're going to contain carbon, hydrogen, oxygen, but less so than in carbohydrates. And what ends up happening is you're going to see that because it has less oxygen than your carbohydrates here, it's going to have more carbon hydrogen bonds, which are going to be it's going to make it overall a lighter molecule. And so we always kind of have this bad, kind of almost negative association, like lipids are bad, carbohydrates are better. That's not the case. All of these molecules are good molecules, especially in moderation, right? And so I don't want you to think of one molecule as a good molecule versus a bad molecule. They're all good molecules, and they're all very important. They just need to be taken in moderation, right? So when we talk about lipids, again, lipids are a necessity for us in terms of, they provide a longer term storage for us. That's not so heavy. If we had to carry all of our energy storage in the form of carbohydrates, we would literally be like twice as big and twice as heavy because carbohydrates are bulky and heavy. So, thanks to lipids, believe it or not, thanks to lipids, that actually allows us to carry our energy in a lighter, more compact form, believe it or not. It's hard to think of lipids that way, right? But that's actually the main purpose, one of the purposes, I should say, for us for lipids. Now, also, lipids are really important because they make up our cell membranes. So we need lipids. Otherwise, our cells wouldn't have a cell membrane because, remember, they make up that phospholipid bilayer of the membrane. Super important, right? So we've got our main types of lipids are going to be triglycerides. So we'll talk about the different triglycerides that we'll take in, especially in foods and things like that. Phospholipids, which make up our membrane. So we need those lipids to make up the phospholipid bilayer of our membranes. We'll talk about steroids. So steroids are going to be this lipid hormone, basically. So it's a type of hormone that's a lipid base. And steroids are going to be things like cholesterol or sex hormones. So things like estrogen, progesterone, testosterone. Corticosteroids, like our cortisol, right, is going to be a steroid. Aldosterone, which we'll talk about in terms of regulating, water in terms of the urinary tract when we get to the urinary system. So all really important hormones, and so they're lipid based. Also, we have these things that we won't talk quite as much about, but eicosanoids are going to be things like prostaglandins that are important for stimulating uterine contractions, especially during labor, controlling GI motility, and promoting inflammation. So again, we'll get into these a little bit more, but all important. Now, what's important is that you all know that lipids, again, are going to be insoluble in water. So lipids, meaning oil and water, right? They don't mix. And so that's because lipids, remember, are a non-polar molecule. So these guys are not polar. And so when you look at it, the reason being is that you've got a bunch of carbon-hydrogen bonds here, okay? So we don't have a lot of oxygen. There's not electronegativity. Remember, carbon and hydrogen like to share equally. So when you think of carbon-hydrogen bonds, I want you to think of non-polar bonds, okay? These are, everything's shared equally and happily, okay? So, I'm not going to play this animation for you just because time, but if you need a little bit more in terms of what's going on here to make our triglycerides, please watch this animation. It's actually embedded in my lecture when you pulled down the lecture. But really quickly, just remember that when we talk about lipids, we have usually a glycerol backbone, and then we have these fatty acid chains. And to remember, in synthesizing this molecule with a glycerol backbone and these fatty acid chains, we are, again, going to see a dehydration synthesis reaction, right? So just like all of them. So our monomers for our lipids are going to be glycerol backbone and fatty acid chains. And then they're put together by this dehydration synthesis reaction where, again, you remove water here and then form a covalent bond in each of these, okay? So
Butter, lard, margarine.
so that's the basic reaction here. Now, when we talk about triglycerides, right? Tri is going to be glycerides are going to be 3 fatty acid chains coming off of that glycerol backbone. And so these, this would be a triglyceride here with these 3 fatty acid chains coming off of here, your glycerol backbone. Now, we'll talk about what makes fats solid versus when they're liquid, which is called an oil. So we call our lipids fats when they're solid and call them oils when they're liquid. And of course, the main functions of lipids are going to be energy storage, which we talked about, right? Okay, we have to have these lipids in this nice kind of compact light form. So it's our long-term energy storage, but also lipids are nice because they provide some insulation, right? And we are a homeothermic organism. We like to maintain a certain body temperature for our enzymes, which we're going to talk about in a minute. So insulation is important, okay? And then also just protection. It provides some protection for, some of our organs. So things like we're going to talk about, like the fact that your kidneys are retroperitoneal. So, some of the fat that you have in your back actually helps protect those kidneys, which are actually outside of the peritoneal cavity. So again, that fat can absorb some of the pressure and blows from a hit. So those are some important functions of lipids. So when we talk about triglycerides, we can talk about saturated fatty acids versus unsaturated fatty acids. So our triglycerides can have chains of fatty acids, because remember they have 3 fatty acid chains of saturated fatty acids. So our saturated fatty acids are going to be where you have all of the carbons here linked to these hydrogen ions via covalent bonds. Okay, so what that means is that they have maximized all of the carbons. There's no carbon-carbon double bonds here. They've maximized how many hydrogens each carbon can bond with, right? So each carbon has bonded with a maximum number of hydrogen atoms. This gives us a nice, really straight chain, really pretty straight chain. And so because of that, when we talk about saturated fats are typically going to be solid at room temperature. And the reason is that these chains pack really nicely and tight together, right? So if you have a triglyceride here that has 3 saturated fatty acid chains, they're going to pack really nice and tight together. And that actually allows that fat to stay more solid at room temperature. So examples of this would be things like animal fats, things like butter, okay? A lot of your dairy will have saturated fats in it as well. Now your unsaturated fats here are going to have 1 or more carbons linked together by a double bond. So it's going to have a carbon-carbon double bond in there somewhere. So just like here, this is an unsaturated fatty acid chain because you have a carbon-carbon double bond. So the carbons are not bound to other hydrogens, right? If you broke that double-double bond, that double-double carbon bonds and made it a single bond, then this carbon could bond to another hydrogen and this one could bond to another hydrogen. So again, that carbon double bond there is going to put a kink in the chain. And that kink in the chain prevents those fatty acid chains from packing nice and tight together, because now you can see it makes the chain all wonky right here. And so that allows the fat to be more liquid at room temperature. So you're going to see this more in your things like your plant oils, such as olive oil, right? And so a lot of these oils are considered to be the good oils, right? For you, it's considered to be heart healthy because your saturated fats are more of the fats that are more likely to cause kind of that plaque formation in your arteries versus, again, your unsaturated fats. Now, not all unsaturated fatty acids are equal, right? So we have these things called trans fats or trans fatty acid chains. And these actually do not occur naturally in nature and are typically produced by an industrial process called hydrogenation. And what happens is they end up forcing hydrogens on, and you end up forming this kind of weird kink in the chain. So normally when you have this kink, for those of you that kind of remember your chemistry, you have the cis formation around that carbon-carbon double bond, and it provides kind of this nice big kink in your unsaturated fatty acids. Well, what happens is on trans fats, they force hydrogens onto these unsaturated fatty acids. And when they do that, it ends up producing this transformation around these carbon-carbon double bonds instead. And when that happens, this will allow these fats to kind of behave more like a saturated fat. So it's still an unsaturated, they can call it, right, an unsaturated fat, although now they're pretty good about marking trans fats on most packaging because we now know they're not healthy, but they behave as a saturated fat. And so they were using them in a lot of products because they're usually solid at room temperature. And so they could use them, but then on the packaging, they could say, oh, well, they could still call it an unsaturated fat. So it didn't look like there was, a lot of saturated fat in that product. So again, then we found out that these were not processed the same way as our regular natural unsaturated fatty acids in the body, and found out that they were actually not heart healthy. So again, we're looking for, and you'll talk more about that. We'll talk more about that kind of later on in 202. Now our omega-3 fatty acids are our healthy ones, right? And so these are the ones that are our natural unsaturated fats, and they'll actually, promote things like, your LDL cholesterol transporters to, that will actually keep the, keep plaque formation from forming in the arteries, right? Prevent that plaque formation. So in a way, your omega-3 fatty acids are your healthy ones. More information than you needed, but I just liked to kind of spice it up, make your chemistry lecture a little interesting here. So now let's look at these triglyceride formation. Again, really quickly, you have the glycerol backbone, 3 fatty acid chains. Boom, you take out a water from each of these fatty acid chains and the glycerol backbone here to do a dehydration synthesis. So it actually takes 4 triglyceride to form. It takes 3 dehydration synthesis reactions to occur, right, to form a triglyceride. And so then you end up with the formation of 3 water molecules when you form a triglyceride. Now remember, phospholipids are also important in terms of lipids. So these are gonna be where you have kind of these modified triglycerides. So instead here you have a glycerol backbone and 2 fatty acids plus this phosphorus containing group, okay, this phosphate group up here. So here's your glycerol backbone, here's your 2 fatty acid chain, and then instead of the 3rd fatty acid chain here, you have a phosphate group coming off, a polar phosphate group. So that the interesting part of your phospholipids here is, as you well know, these fatty acid chains, right, are non-polar. Lots of carbon-hydrogen bonds, non-polar. But the crazy part is this phosphorus group up here is very polar. So again, lots of here, if you look, phosphate, lots of oxygens around there, very electronegative, so very polar. So what ends up happening is you have this polar head region right up here, right? That's going to want to interact with things like water molecules. And then the tail region, which is non-polar and not going to be attracted to water molecules and actually repelled by water. So again, the head is your polar region up here, that phosphorus, that phosphate group up there, and is attracted to water, and then the tails are non-polar and repelled by water. So again, this is really important in that phospholipid bilayer structure, because what ends up happening is the inside of your membrane, if you think of it like a sandwich, right? So you've got a double layer of these, phospholipids, phospholipids with the tails pointing to the inside, and it's going to form this kind of sandwich middle here. The peanut butter and jelly is going to be all of these, hydrophobic tails. So these non-polar hydrophobic tails that are not going to want to interact with things that are charged. And then on the outside and the inside here, so this would be the outside of the cell, from the outside and the inside of the cell, you're going to have these, polar heads with those, polar phosphate groups that will actually interact with water. So it really makes the membrane, gives it this perfect structure. So it's really, really important for the structure of the cell membrane. So again, this just kind of gives you exactly what we talked about, the glycerol backbone, the phosphate group here that's polar, and then the non-polar tails that are made of the fatty acid chains. Now we got steroids as well. And don't worry, we'll talk more about the phospholipid bilayer, of course, when we get into the cell lecture and membranes. So next we're going to talk about steroids. And steroids are, again, lipid-based hormones. They're going to consist of these 4 interlocking ring structures here. So again, there's still going to be a lot of carbon-hydrogen bonds in here. And so some of our common steroids that we're going to talk about are cholesterol. Believe it or not, vitamin D is based on a steroid here. So vitamin D is technically a steroid. Steroid hormones are going to be things like our sex hormones, so things like testosterone, progesterone, estrogen. Corticosteroids, so things like cortisol, aldosterone, which we'll talk about again in terms of the urinary system, and then bile salts. So all of these things are very common steroids, important steroids in the body. The most important steroid is cholesterol, and that's because this cholesterol here, as you can see here, is the building block for many of these other bigger steroid molecules. So it's a building block for vitamin D, for steroid synthesis, and bile salt synthesis. And so you can see, you'll have oftentimes this cholesterol group, and then, you can have other things attached to that cholesterol. So this, believe it or not, cholesterol is actually quite important in terms of the structure of plasma membranes. And it's actually one of the major reasons why our plasma membranes are more fluid, is thanks to these cholesterol molecules, because they kind of prevent these phospholipids from being too packed in, too tight, and actually helps to give our membrane its fluidity. So it actually helps it to keep its fluidity as well, in terms of, when we have some temperature changes. So cholesterol we'll talk a little bit more about, but very important in terms of cell structure in the plasma membrane. So again, here is our kind of simplified structure of a steroid. We got to have that cholesterol again, those 4 interlocking hydrocarbon rings there being our basis for all steroid formation. And then you have, again, different side groups coming off. Eicosanoids, we'll talk just briefly about, we won't talk a whole lot about them in Unit 2.1 and 2.2, other than just kind of their role in different things. So there's many different eicosanoids. Again, we'll just talk about a few of them. They're derived from this fatty acid called arachidonic acid that's found in many of our cell membranes. Not really important that you know that, but what I do want you to know is that there are some important eicosanoids, and the main ones are prostaglandins. Our prostaglandins are a big one. You'll hear prostaglandins quite a bit in terms of they have a major role in blood clotting. So, for example, we'll talk about thromboxanes. Again, they're really powerful vasoconstrictors, very important in terms of, again, the clotting process. We'll also talk about prostaglandins in terms of their control of blood pressure. They're also really big in terms of promoting inflammation. Very important in terms of stimulating uterine contractions for labor. And then they also help to control our GI motility. So, they're a smaller group, but still also very important. So prostaglandins are a big one. I want you to know that prostaglandins are an eicosanoid. And so they're in this category of lipids because of that. So they have this lipid-based, again, structure. And what's kind of interesting is that there's this recent article. I posted a link here for you, for those of you that are interested. It was really interesting. Kind of more recently, they found out when talking about SARS-CoV-2 and COVID, that SARS-CoV-2 infections can actually, sometimes lead to, depending on the individual's response, some severe tissue damage. And when that happens, a lot of cell debris is released. And so the actual, both initial primary infection they found, as well as the accumulation of a lot of the kind of cell debris from, the destruction from the virus, the tissue damage from the virus, can actually stimulate this, what we call endoplasmic reticulum stress response. And what ends up happening is they found out that upregulated these kind of inflammatory enzymes and then produced what we call an eicosanoid storm. So that produced then a bunch of prostaglandins, all kinds of things, leukotrienes, which is another eicosanoid, thromboxanes to be released, which became part of the problem with some of the clotting issues. And the severe vasoconstriction there. And then these then were what induced the cytokine storm. So and that cytokine storm then, when these certain cytokines were released, they ended up causing this really widespread inflammatory response that's really did the damage in terms of, some of the organs like the heart and the lungs. So anyway, more than I probably wanted to get into. So you can fast forward this if this is just too much, but I just thought it was interesting, and it was an interesting way to kind of help you see the role of eicosanoids. Really interesting to see there's this kind of recent research on their role in terms of this widespread inflammatory response to SARS-CoV-2. So, but I'm not going to test you on that, just kind of a side note in terms of that. Proteins, really quickly. So proteins comprise 20 to 30% of the cell mass. They have this kind of very varied function in terms of kind of the most varied function of any of the molecules. and their structure is very important. We'll talk about that. They are going to make up enzymes, which are important to, all of our reactions in our body, and they're important for things like muscle contraction. Of course, here, when we talk about our monomers, here we are back to our monomers and then our polymer, right? So our monomers are going to be the amino acids, these basic building blocks, the amino acids. And then as we put those amino acids together, via peptide bonds, so we take one amino acid, peptide bond it to another amino acid, then we're going to start to form these polymers. And so when we form the polymers, those are going to be our proteins, okay? So all proteins are going to be made from only a group of 20 amino acids. So all the proteins that we have in our entire body are made up of just 20 different types of amino acids, and how we combine those amino acids and the order that we combine them in is going to be what's going to define one protein from another. So again, we're going to join these amino acids by these covalent bonds called peptide bonds, right? And remember, each amino acid will have an amino group and an acid group, the carboxyl group here. And then, of course, they're going to have an R group. So you have the central carbon attached to an amino group, attached to a carboxyl group, attached to a hydrogen here, and then attached to an R group, the side chain. And that R group, there are 20 different ones. And that's what makes each amino acid different from each other, is this R group here. Okay, so, and again, of course, I have my nice, funny cartoon for you there to keep it real, keep it interesting. So again, here you're going to see we've got an amino acid here with its amine group attached to a carbon, attached to a R group here, attached to a carboxyl group here. So one amino acid here. And if we are going to build a peptide bond between one amino acid and another amino acid, again, you're going to see this reaction over and over again. Here we go. Dehydration synthesis, right? To build that peptide bond. So again, we're going to pull out of water and form a peptide bond between that carbon and that nitrogen here. Okay. Now, of course, when we do that, then we make a dipeptide, two peptides together. Okay. Now, if we were to break that peptide bond, okay, we would require water, right? And then we'd have to lyse that water and put a hydrogen on one end and a hydroxyl group on the other. And we would call that process hydrolysis. So to break down our, proteins, we require water. So you need water to break down any of your molecules. So for example, if you're breaking down a bunch of carbohydrates and proteins because you just ate, you require a lot of water to do that. Because to break these bonds, again, we have to do this hydrolysis reaction. So just kind of interesting to keep that in mind, right? With water is important for even things like digestion. So when we talk about proteins, protein structure is extremely important, and it really helps to determine the shape and the function of our proteins. So again, just as kind of a review, right, from 181, we have the primary structure of our proteins, which is just this linear sequence of amino acids, right? What order are the amino acids in? Do you have a leucine and a proline and then an alanine? I know you don't have to know those amino acids, but that's just an example of like a linear sequence of amino acids that we would have, right? So that's our primary structure. So here's our primary structure, right? Just simply that linear sequence of amino acids. You've got amino acid 1, here amino acid 2, amino acid 3. That's our primary structure. Now our secondary structure is how those primary amino acids are actually going to interact with each other. And they're actually going to form some hydrogen bonds. So they can either form a helix, okay? So these amino acids will form hydrogen bonds in a form that will coil that string and form a helix, kind of resembles a spring. We call that an alpha helix, or it can cause it to form these accordion ribbons, what we call a beta pleated sheet. So let me show you this. So here again, what will happen is you'll see these little dotted bonds here are actually going to be hydrogen bonds that are forming. And so between the different, R groups, you can, depending on what these R groups are, they can interact with other amino acids based on whether or not they're polar or not, right? And if they're polar, they can form these hydrogen bonds. So here in this case, hydrogen bonds are formed to cause this linear chain to form this helix, which we call an alpha helix. Now here, they can also cause them to form these what are called beta pleated sheets, where it kind of zigzags back and forth. So here the chain would kind of fold back on itself and zigzag based on how those R groups are interacting with other amino acids. So that's called our secondary structure. And our primary structure, remember, really determines what our secondary structure is going to be, because the R groups are a big reactive group there. And then our tertiary structure is where you'll see then we get even more folding and interactions between the chains. So you'll get interactions between your alpha helices and your beta sheets here. And that will fold kind of back on itself even more, folding up that molecule to a kind of more compact or even globular form. So that's what we call tertiary structure. And then quaternary structure is when you get two or more polypeptide chains, each with, again, it already has this one has its own tertiary structure, and this one has its own tertiary structure, and then they'll combine to form a functional protein. And so, you can get two polypeptides, sometimes four polypeptide chains coming together to form this, big molecule. And it'll give it this really complex structure, which oftentimes is what gives that protein its function, is that structure. Okay. So those are the four different types of folding. So again, tertiary here is how the secondary structures are going to interact. The alpha and beta pleated sheets interact with each other. And then quaternary structure is when you have, again, those two or more polypeptides that have their own tertiary structure and they're interacting with each other. Okay. So then we have what are called fibrous and globular proteins. And this is kind of important because we'll talk about the different purposes of that. So our fibrous structural proteins, okay? So again, these already have their own structure. Our fibrous structural proteins are going to be kind of more strand-like you see here. And they'll be oftentimes water insoluble and very stable. A lot of these, again, most of them have tertiary or even sometimes quaternary structure that give them, again, even this fibrous chain-like structure. And so what's important about many of your fibrous proteins are that they're going to provide kind of more mechanical support and tensile strength. We're going to talk a lot about these in terms of things like connective tissue when we get into that. So you're going to see a lot of these fibrous proteins in your connective tissue. So things like elastin and collagen, those, you're going to find in your connective tissue. Also keratin, the stuff in your fingernails. Also, again, a fibrous structural protein. So really important, also certain contractile fibers that we're going to talk about in terms of muscle contraction. Also, again, really important fibrous structural proteins. So that's an example of our fibrous structural proteins. And we also have these globular proteins, right? And these globular kind of more functional proteins, so we call these ones the fibrous structural because their purpose is going to be more structural, but the globular proteins are going to be more functional. And these are going to be a lot of the things that make up our enzymes. They have a very special structure, and that's because they have an active site that needs to bind to a substrate. Okay. So these guys have a more complex structure. They're oftentimes, again, water soluble. They're sensitive to environmental changes. Again, they have these tertiary or quaternary structures. We'll talk about how they're sensitive to environmental changes. Things like temperature can denature them, things like that. Acids, really basic areas, basic environments can denature them. They have these very functional regions as well that are our active sites. So again, maintaining this structure is very important. And some examples of these are, hormones, molecular chaperones, And then, of course, enzymes. So again, antibodies and enzymes are really easy to picture how important that shape is, that overall structural shape is. So we'll talk about enzymes in just a minute here. So of course, proteins, the challenge with proteins is that they're subject to denaturation. And that's the fact that proteins can be denatured. So globular proteins can unfold and lose their functional 3D shape. And that's really important to keep in mind, because when we talk about enzymatic function and enzymes, and remember, every single metabolic reaction in your body requires an enzyme. And so these enzymes are going to be very picky at what type of environment they're stable at. So fibrous proteins tend to be more stable, but globular proteins are less stable and more likely to denature. And then when they do denature, those active sites, like the ones on enzymes, become deactivated. So a lot of times, again, decreased pH, increased acidity can cause a protein to denature and break down, unfold. Also increased temperature, which is easy to picture, right? When you think about cooking an egg, you can't undo cooking an egg. So usually, again, usually reversible if normal conditions are restored. But if, again, extreme changes happen, they're irreversible. So for example, again, you can't undo cooking an egg. Once you denature the protein, you can't go back the other way. You can't refold those proteins. They're too, they've lost too much of their structure to be able to go back. So now this is where we get into the real meat of proteins, is enzymes. And enzymes, again, are these globular proteins, and they are our biological catalysts, which I know I've talked a lot about already. So these guys are going to increase the speed of chemical reactions without actually getting used up in the process. So that's the great part, is enzymes just help a reaction to occur, and then when they release the products, then they can go on and bind another substrate. So that's the beauty of enzymes. Now, how enzymes work is they, again, aren't actually getting involved in the reaction themselves. They're actually just helping to lower the energy, what we call the activation energy that's needed to initiate a chemical reaction. So again, this is just going to help speed up that reaction. It allows for millions of reactions to occur per minute. So when we talk about this, like I said, all of the reactions, the metabolic reactions that happen in our body have enzymes. Most of them have enzymes that are required for that reaction to happen, because it would just happen way too slowly, right? To keep up with the needs of our body. So we need these enzymes because we need these metabolic reactions to happen at a much quicker rate than they would on their own. So when we talk about enzymes, kind of important, not all of this is important, but just terminology. But the protein portion of the enzyme is called the apoenzyme. And then there's something called a cofactor or coenzyme. So sometimes our enzymes require either a metal ion cofactor or a coenzyme, which is sometimes just an organic molecule, oftentimes a vitamin. So this is why sometimes we need certain metal ions or certain, again, vitamins, is because they are needed for the function of an enzyme as either a cofactor or coenzyme. So enzymes also, as you well remember, are very specific because of their folding. They're very specific. The protein portion, the apoenzyme portion, has an active site that will only bind a very specific substrate. And the names usually end in ACE, A-S-E. That's how you can recognize you've got an enzyme, and are often named for the reaction that they're catalyzing. So, for example, when you're talking about, hydrolases, that's the enzyme that's going to add water during a hydrolysis reaction, okay? Or oxidases, that's an enzyme that's going to oxidize the reactants by adding either oxygen or removing a hydrogen, okay? So those are enzymes that are going to help those types of reactions occur. So just to give you an idea. Or like something like lactase, lactase is an enzyme that's going to break down that that bond, right, that carbohydrate bond that's formed between that disaccharide, lactose. So again, just kind of how a little bit on how enzymes work. Enzymes, remember, are just going to lower the activation energy, which is the energy needed to kind of initiate that chemical reaction. So that's why I like this analogy here. So enzymes are just kind of lowering that energy barrier that's required to make that reaction occur. Enzymes allow chemical reactions, again, to proceed quickly at body temperatures. So 3 steps that are involved in our enzyme reaction. Again, the substrate needs to bind to the enzyme's active site, and then this temporarily forms the enzyme substrate complex. Then that complex is going to undergo this rearrangement of the substrate, kind of torquing it and putting it in a position where maybe the bonds want to be broken or maybe bonds want to be made, depending on what type of reaction you have. And that will result in the final product. And then eventually that product is released from the enzyme, and the enzyme is totally unchanged and can go on to bind another substrate. Okay. So again, this is just kind of that analogy of lowering, again, that activation energy. Down here is the analogy, sorry. And this is showing kind of the activation energy that might be required for a certain reaction to occur. And then with the enzyme, you know, that activation energy is significantly lowered. Okay. This just shows you, again, the active sites. So maybe here, in this case, this enzyme is helping to form a peptide bond between 2 amino acids. So again, it would be very specific for the binding of these 2 amino acids. When they bind, this forms the enzyme substrate complex, and temporarily, right, temporarily, and then that enzyme substrate complex can undergo kind of its own internal, or, you know, changes and arrangement to allow, again, energy to be absorbed for that bond to be formed. And then water can be released, and boom, the enzyme will release the product of that reaction. And in this case, it would be a dipeptide. Okay, so that's just kind of how the basics on how enzymes work. Our last molecule that we're going to talk about is nucleic acids. And nucleic acids are going to be the largest molecules in the body. And these polymers are going to be made up of our nucleotide monomers. So our nucleotide monomers, each nucleotide, remember, has a nitrogenous base, right? Okay, attached to, so a nitrogenous base attached to a pentose sugar and then attached to a phosphate group. So that's what makes makes up each of our nucleotide bases. So there are 2 major classes. We have the deoxyribonucleic acid and then the ribonucleic acid. So here you can see, remember, deoxyribonucleic acid, or DNA, will form a double helix and has the nucleotide bases cytosine, guanine, adenine, and thymine. And then our RNA, right, is single-stranded, and will have cytosine, guanine, adenine, and instead of thymine, it will be replaced with uracil. Okay, so the nucleotide bases for each. So again, our DNA is important because that holds the genetic blueprint, right, for the synthesis of all the proteins in our body, right? So it is the big genetic blueprint that codes for the proteins in our body. The double helix, as you guys probably remember, and that double-stranded helix is going to be located inside the cell nucleus. Again, nucleotides for DNA are going to contain a deoxyribose sugar attached to a phosphate and then one of the 4 nitrogen bases. So either adenine or guanine, which are purines, or cytosine and thymine, which is our pyrimidines. So those are the basics for DNA. And remember, when we talk about DNA and base pairing to form this double helix, what forms this double helix is going to be the bonding of the nitrogenous bases from opposite strands. And so just as a reminder for our complementary base pairing rules, you know, you're always going to have, let me just draw this in here. You're always going to have, you know, an adenine is going to have to pair with a thymine. So if you have an adenine over here, okay, then it's going to have to base pair with a thymine over here. And then guanine is always going to base pair across from cytosine, okay? And that's what's going to form that nice helical arrangement. So if you know you have a cytosine here, then it's easy, you know that there's a guanine here, okay? Or if you have a thymine here, then you know there's an adenine here. And so this is going to help us to form that nice double helix, right? And of course, back to our hydrogen bonds here too. Sorry, I forgot to mention that the bond between these is going to be a hydrogen bond. So it's a hydrogen bond between the adenine and the thymine there, and then the cytosine and guanine there. It's all hydrogen bonding. And this just is nice because you can see here that each of these, you know, nucleotides consists of this nitrogenous base attached to a deoxyribose sugar attached to a phosphate group. So you can see that. Now, when we look at RNA, remember that RNA links DNA to protein synthesis because, again, from your DNA, right, you're going to make this kind of copy of the code in the form of messenger RNA and then send it out of the nucleus, right? Because we don't want to send out our DNA, our hard copy of things that, you know, we want it to stay safe inside the nucleus. So our RNA is a single-stranded kind of linear molecule, right? And it's active mostly outside of the nucleus. We keep our DNA in the nucleus, and our RNA will send outside the nucleus. It contains a ribose sugar, okay, instead of the deoxyribose sugar. And thymine is going to be replaced with a uracil, okay? So, you know, instead of base pairing, you know, when you where you'd have a thiamine, uracil would be replaced. So the three varieties, and we'll talk about, again, the process of going from DNA to RNA in the next lecture. So I'll talk on how we get messenger RNA in more detail. So there's the three varieties of RNA that are going to carry out our DNA orders. Just to kind of mention them, there's messenger RNA, transfer RNA, and ribosomal RNA. And again, we'll talk a lot more about those. And then our last nucleic acid that we have to talk about is, of course, adenosine triphosphate, okay? So again, we have this nitrogenous, just like all of our nucleic acids, our base, you know, our base nucleotide, our monomer here is going to have a nitrogenous base attached to, in this case, a ribose sugar, and then 3 phosphate groups. And that's what makes up our ATP. So ATP is technically adenosine, right? Triphosphate. So the adenine and the ribose make up your adenosine, and then triphosphate, 1, 2, 3. So ATP, right, ends up being our like energy form of the cell. And so this is the chemical energy that's released when glucose is broken down through that, remember that series of reactions that we talk about in terms of the Krebs cycle. And ATP is going to, you know, directly power all the chemical reactions in our cells, right? So our ATP is going to be our chemical energy of the cell. It's the usable energy needed by all of our body cells. And again, it's that adenosine, adenine, ribose, adenosine triphosphate. Again, that's just kind of another depiction of the adenosine triphosphate. And this is just showing these terminal phosphate bonds between these 3 phosphate bonds that are actually the really high energy bonds. And so as you break those bonds, energy is released. And that's the energy that we're actually using to do work. So when we lose one of these phosphate bonds, right, we go from adenosine triphosphate to adenosine diphosphate, and we'll release a phosphate group and energy as a byproduct. And that energy that's released is what we're using to do work for the cell. Okay, and so some of that work that we do for the cell is transport work. So we'll talk about that in terms of bringing things into and out of the cell. Some of that energy is going to be mechanical work in terms of doing things like muscle contraction. And then some of that energy is going to be chemical work. So again, it'll be used to help, make bonds between two reactants and things like that. So we'll get much more into that into our next lecture. So please let me know if you have any questions on this. I know that I went through some of this quite quickly towards the end, and I also know that it was a little bit long. Like I said, I made these lectures so that you can go through at your own pace. Not everybody will need these full lectures, so you're welcome to truncate them as you need. And then if you need more detail, it's here for you. All right, please let me know again if you have any questions. And as always, I'm available through all the forums. You can, again, contact me through email, through Canvas, and then I can always set up a time to chat with you.